Hi everyone, welcome to this video about DNA structure and protein synthesis. So what we'll be looking to do in this video briefly is to explain the structure of DNA and then how we go from a molecule of DNA to protein itself through transcription and translation. This is aimed for GCSE students. There is a video on this for A-level students with a bit more detail if you're interested. So let's start with the DNA structure itself. So your DNA molecule is basically a molecule that contains all the instructions for life, as you know from what we've learnt in year 9. So DNA itself, it stands for deoxyribonucleic acid. So it's a form of a nucleotide, and a nucleotide is the monomer, and a polynucleotide is what it's called when it's joined up as a chain to make your deoxyribonucleic acid. Now, the DNA itself has a double helical structure, which basically means it's got two strands twisted around each other. It's made up of three parts as nucleotide. It's made up of a phosphate group, it's made up of a pentose sugar, and it's made up of nitrogenous bases. Now, these four bases are adenine, thymine, cytosine, guanine, and they have a special way of pairing up. So adenine will always ever bind with thymine, and cytosine will only ever bind with guanine. At GCSE, you don't have to worry about why that is, but basically it's because of the number of hydrogen bonds they form together. So the hydrogen bonding happens between the bases on one strand to the bases on the other strand. So what you'll have is basically like a ladder type structure. And that ladder type structure will twist around each other. So this is why I call this a DNA helix. Okay, so that's your basic structure of DNA. Now, what we need to think about is how do we go from these nitrogenous bases, from the order of these bases, to a protein? So the first thing that needs to happen is we need to unwind a part of our DNA. Protein synthesis actually starts in the nucleus. So what happens with this? We copy a section of DNA called a gene. That DNA unwinds, an enzyme causes it to unwind, and another enzyme called RNA polymerase actually makes a copy of the gene in the form of something called mRNA. So that's messenger RNA. And so unlike DNA, it has this uracil rather than thymine. That's one of the major differences. It's also single-stranded as opposed to double-stranded. And it's not deoxyribose, but ribose as the sugar that it has. So the advantage of this is mRNA is small enough to be able to leave the nucleus and go to our ribosomes where we're going to actually make our protein. So exam tip is the DNA sticks in the nucleus, stays in the nucleus, but mRNA moves out to the ribosome. So once you've transcribed that DNA, we now need to translate it. So as I said to you before, the mRNA actually leaves the nucleus and comes to your ribosome. So your ribosome will hold your mRNA in place, and it reads the mRNA in something called triplets. So that's basically three bases as a codon. So this is your codon here, as the three bases are read. It doesn't overlap, so it will read three, then move on to the next one. And in ribosome, the mRNA meets a molecule called tRNA. And the tRNA is basically a codon with an amino acid attached to it. And what happens is this codon will then read the triplets, the codons, and it will attach amino acids together. And those amino acids then are joining together to actually make your protein. So the protein will finish into a specific shape, and we'll look at that in a little bit. The key example here is codons are always read in groups of three. For example, AUG codes for an amino acid called methionine. That's your start codon. It will always just read in three and then move on. So the importance of this is that proteins are essential. They make up everything. They make up enzymes, hormones, structural proteins. So that can be the enzymes that speed up reactions in your gut or your digestive system. So, for example, amylase breaking down starch. You've also got hormones like insulin, which control your blood glucose levels, and glucagon, which works in conjunction with insulin. You've also got structural proteins like collagen in your skin. You have proteins in plants such as ribosco, which is the basis of photosynthesis. Did you know that a mutation in DNA can lead to a change in protein, and that's how some of the genetic diseases occur? But we'll have another video on that later. So, as a quick summary, DNA is a double helix made up of nucleotides. We've got four bases: adenine, thymine, cytosine, guanine. Adenine binds with thymine, cytosine, and guanine. Transcription happens in the nucleus where you change the DNA, a section of DNA called gene, into an mRNA. mRNA has uracil rather than thymine and single-stranded, so it can leave the nucleus. And translation happens in the ribosome where tRNA helps to form a protein by bringing amino acids into place. So an exam question, for example, you could get on this is a section of DNA has base sequence TAC, GGC, ATA. What would the corresponding mRNA strand be? Pause this video and have a go. So, if the base sequence initially was TAC, GGC, ATA, for mRNA it'd be AUG, CCG, UAU. Remember, in mRNA you have uracil rather than thymine, and it'll be the opposite, so complementary bases, adenine with thymine, cytosine with guanine, or adenine with uracil in this case. Always double check your base pairing and remember uracil in RNA. That's it for today. I hope this helped you understand DNA structure and protein synthesis. If you found this useful, don't forget to like and subscribe and comment below with any questions. Thank you.